A human genome can now be sequenced for $100, whereas the original human genome that was sequenced cost close to $3 billion and took over 13 years, whereas now we can do it in a single day. So what does this mean? Why is this a big deal? And what does it allow us to do in the future? Well, that's what we're going to talk about today. My name is Rob.
And I'm Stephan. We are two biological data scientists passionate about using data, science, and tech to become our best selves. And this is episode 13 of the Fit for Science podcast, where we usually discuss wearables, quantified self, data, and how we apply all of those to our own life. So sequencing has crossed a cost threshold of $100. It has become cheaper and cheaper over time in the last 20 years. And this could actually have huge implications for personalized medicine. And that means how we prevent some of the biggest killers in the global north, like heart disease or neurodegenerative diseases such as Alzheimer's, which have a genetic component. But let's take a step back first. What does it actually mean to sequence your genome? And here I think we are two good people to talk to or to talk about this, as when we talk about biological data science, what we often mean is actually the analysis of genomic data, and that's part of our day-to-day job, right?
Yes.
So let's try to define what genome sequencing actually is, and also how it differentiates in terms of end consumer products that people often think about in whole genome sequencing, or it's not whole genome sequencing.
Yeah, maybe let's start with the most famous company, 23andMe. So many people will have analyzed their genome with 23andMe. And that means getting some feeling for the code of your DNA. So every cell in your body has the exact same DNA, and we can read this and record it to see your genome. Now, there's whole genome sequencing, and there are these companies like 23andMe that don't actually measure all your DNA. They select a certain amount of spots on your DNA, let's say 100,000, and they read on these spots what is your code, but they ignore the vast majority of your genome. Whereas with whole genome sequencing, you don't pre-select those spots. You're just like, I'll read everything that there is and try to piece together how they actually form your entire genome. So this is a specific technology that was developed basically where you cut up your DNA, you read all the separate parts, and then you try to puzzle back together how those parts are in order. This is very much also a computer task that we have optimized over time. And now we can do this very efficiently. So instead of just measuring the most important spots, which most important is kind of not completely random thing, but there's decisions to be made there. We can actually measure everything unbiasedly, and this is actually in principle for most cells of your body. This will be the code that it always has, except for certain mutations that are actually always bad. I don't think there's a typical scenario where there would be any positive mutation while you are already alive. And this then provides basically a blueprint for your entire DNA.
Yes. I think a good example or analogy that works well is always that the DNA is, as you said, in every cell the same, but it's a book. It's a really big book. And maybe to differentiate whole genome sequencing from what these some end consumer products do, this 23andMe, when it's called genotyping, if you then really look at it, they determine your genotypes. The whole genome sequencing is really, you go in and write the whole book, or you go to the library and lend the whole book. So you have every single page, everything that is written in there. And what they do is they say, actually only, as you said, 1,000 chapters matter, but the rest we ignore. I've looked up the numbers just to give it a bit of a context. So, the DNA in general is consisting of 3 billion base pairs, roughly. That means 3 billion letters, so to speak. And that's really the biochemical code how genes and then hence proteins, and which are the functional unit of life, are encoded in your DNA. And then compared to that, these 1,000 chapters that we land only in when we use something like, or have used something like 23andMe, it's actually only roughly 0.02% of the genome. And as you also pointed out, there had to be decisions made, and the decisions made are based on our current knowledge. Hence, you know already something about these addresses in the genome, so the chapters, and that's why you want to read them out. But actually, if someone, and genomics is an extremely young field compared to other sciences, or natural sciences. If you find something out about another address that is very important for a disease that is still developing in terms of that we understand how it works, like Alzheimer's, you cannot just go back and check this chapter because you have learned only 1,000 chapters. And if you need a new one, you cannot check it. But with whole genome sequencing, if you really read everything, then you can actually go back because now you have actually recorded the whole book and you can really go to the page and see what kind of text is there in your case. Maybe one more thing for traversing this, there's 30x and 100x whole genome sequencing. It just means that every base pair on average was sequenced 30 times or 100 times, and it gives us more confidence that each of these base pairs was correctly recorded. I think that
maybe
suffices.
To simplify it,
yes.
Basically we have a scanner that goes over each line of the book, but there's some uncertainty there. Maybe it accidentally reads a B where it should have been an 8 or something like this, but 90 whatever % of the time it measures the correct letter or number. And by doing this a lot of times, you get more confident that what you read was correct. And you want a lot of confidence because you're reading so many letters that mistakes are inevitable, even if it's only 0.1% of the time that you're getting it wrong. If that's over billions of letters or millions of letters, you can imagine that there will be a lot of mistakes, so you need to read them a lot of times.
And sometimes a mistake of one letter is actually quite consequential.
Yes.
It can be. Usually not, but it can be, right? So
and
that's quite important.
Exactly. And in some scenarios, there might also be a bias that certain chapters are read a bit more, so you have more confidence in them, but then you also need to make sure that the chapters that are read a little bit less are also measured with a lot of confidence.
Yes. Exactly. So I think now we really defined what sequencing is. It's basically this biochemical and bioinformatics measuring and quantifying of one's genome. So how the DNA basically structured, and it's a one-time measurement in theory per person. That's very important, I think. And this has now become cheaper. And you said in introduction, it has cost, the very first one was a multi-year, 12-year project and cost approximately $3 billion. This is really hard to quantify because there were so many researchers and institutions and countries involved that there's always this estimate thrown around. So it's really, really hard. But now a company called Element Biosciences have a new system called Vitari, and they announced if you have this system, so the acquisition cost is not included. These systems or machines cost quite a lot of money, like in a million, like I think roughly a few million, or a little bit more than 1 million sometimes. Then you can actually sequence one whole genome at the cost of $100. And yeah, this then led to some headlines, what it might do, right?
Yes. And there's even the suggestion that if this gets cheaper over time, it might actually be cheaper to resequence it if you need it than to store it on some storage somewhere, depending if storage gets cheaper or not, because this is usually a decently sized data file. But that's a side note. If this gets a lot cheaper, then it might be easier to just measure your genome each time your doctor needs it, because what are the odds they need it again this year versus storing all the data from the whole population.
Just to put in context, maybe it's a few hundred gigabytes, right? If we just now, we just estimate as well. So this is just guesstimations. And I don't know, you pay for Google Cloud for 100 gigabytes for a year, $24, right? Or 24 euros, whatever. Just to put it also in terms of cost into perspective, I've looked up, and this is also guesstimations, but an MRI, which only gives you a snapshot, for example, if of your knee or whatever, why you need an MRI, costs between $400 and $3,500 in the US if you pay out of pocket even more. So 1,000 to 5,000, which is wow, because I mean, the human genome has so much more information in general about your body, how everything is built in you, than, for example, an MRI. But obviously, you need an MRI for the snapshot because maybe something happened and you want to know how it is now. But just to put it into perspective, both the storage costs, it's a really good point, and also how much actually other stuff that you might have gotten already, or an X-ray, costs for the medical system.
Yes, and this data is quite useful from the genome. And here maybe it's good to define what kind of diseases you can detect with this. and why for some diseases maybe this kind of genotyping, so the very cheap, just look at a certain location is enough, and for other situations the sequencing is a lot better. So the classic, I think, original way we often used genetics is by looking at a single location where we know if you have a mutation there, which is probably very rare, only whatever 1 in 100,000 people has this, then you have a major disease because we know there's something wrong, probably already from childhood onward, and people wanted to explain why does this person have this disease. So
Like
this
a
kind of monogenic Mendelian type of disease. And this could be quite cheap to do in other ways. But the moment you want to move beyond that and a disease is not caused by a single location on your genome, but it's a combination of factors that can even interact in very complex ways, then you almost need, if you want to look at a lot of those, a whole genome to be sequenced, or at least it becomes the cheapest way of doing it.
Yeah, I think we've recently discussed in another episode how these genome-wide association studies are done, and you have done actually something similar at work recently, and also then something related we will introduce now is polygenic risk scores, and this is very closely related. So genome-wide association studies is when you have a lot of those genomes of individuals and have corresponding trait information. A trait can be a disease, but it can also be a behavior or something else. So everything that describes anything, or a height could be a trait. And then you associate it to find out which addresses, which locations in the genome, and what is there in kind of term of sort of letter is associated with that trait. And if you do this with enough genomes, you can find statistically significant locations that do predict to some degree the trait. And what you can do then is determine a polygenic risk score. Poly just means many, and genic means from genes. And risk score in this way is negatively phrased, right? Because being tall, it might not be a risk, it might be desirable. I don't know, but or I do know. But polygenic risk scores can be then, hey, we can quantify actually compared to a control group or the general population, how much more likely is it for you to have the certain trait given your genome, right? And this is something that has been around for a long time. And you have been recently calculating this, right, for a study that you're involved in.
Yes. And I think here we should also make clear again that what the difference is between a gene and a mutation, or how these polygenic risk scores work, because we all have the same genes in terms of the gene exists, but the exact code of that gene can differ a tiny bit. Mm-hmm. And that tiny small effect in the code usually slightly affects the functioning of the gene in a way. Mm-hmm. So we both have a gene called, let's say, ARP1. I don't know if this exists, but these genes always have short names. And maybe in location 1001 on that gene, you have transformed an A to a T or whatever. And that could have a small effect. Usually, these are not deadly effects because then you wouldn't be born. So in the general population, because your parents had certain variants, we call them, so mutations of locations on genes, you will get a combination of those. But we somehow in the common tongue, often we say a gene is mm-hmm. different, but we still have the same gene. Yes. It just functions a tiny bit differently.
Some people even say, I have the gene,
exactly.
Right? Which is also just a colloquial way of saying I have this variant that is associated with a certain trait that was found. I think a very famous example is BRCA genes, BRCA, the breast cancer related. So they're related to the trait of breast cancer. And it was found because there's such a strong predisposition. If you have these genetic variants, the increased likelihood of developing breast cancer over lifetime. And there, I think it's a really good point to, again, I mean, we discussed it, I think, 2 episodes ago, but nurture versus nature. What do I mean?
So nurture is your environment and how that influenced you, and nature is your genetics.
Yeah. And genetics is exactly what we are talking about today. And also when we say polygenic risk scores or association of risk or predisposition, what we always mean is, I like this term, genetics load the gun, but epigenetics, which means lifestyle and environment, they pull the trigger. And that means there's only an increased chance, a probability that you have more compared to average, right? That does not mean that someone who has a very low number cannot get it. And that doesn't mean that someone who has a very high number must get it. It's it's probabilities.
Yes, and that is in contrast to those very usually monogenic diseases where if you have that mutation, you will get the disease. Yeah. There are those examples, and that's not what we're going to talk about today, because the reason we're using multiple genes or multiple locations on those genes is because we cannot predict it without using all of them.
It's somewhat the olden days of medical genomics. It was like, okay, first let's look at the stuff where we have this one-to-one mapping, and now we get into this age of looking at the combinations that yield a trait.
Exactly, and it will be that combination of the polygenic risk score with your environment that in the end determines do you get the disease or maybe the positive outcome. And a simple way of looking at it is just saying you sort of add up your risks over the different genes, and then you get a total risk score. It's a little bit more complex, but you can sort of see it like this.
Yeah, it's basically, I think it's very close actually to reality. And once our boss actually said, with genome sequencing, it's like if there's something seriously wrong and you're already around your 30s, you would know.
Yes.
That reflects what you said with there's this monogenic disease, and then this is really for the serious stuff, and the other stuff informs. But that means now that raises the question, okay, now we can sequence this. This is very cheap. Then we can determine these polygenic risk scores. Why do I not have, as a normal human that walks around, my polygenic risk scores determined by, I don't know, my GP or whatever? Why is this not a thing? And also the question is, maybe that not everyone wants this, because as we kind of emphasized, it's probabilities. But as we've seen with the corona pandemic, but in general, we have not a very good grasp as humans of probabilities. And also we cannot really well navigate it. And often people are actually, they don't like to know this because you cannot take away knowledge again, right? If I tell you now you have a 50% chance of something bad happening, and you have not mentally prepared and agreed that you want to know this knowledge, who am I to basically up your day or your week, or I don't know how long it takes to digest this information. So yes, there is a significant risk of anxiety induced, right? And the problem, I think, until now was that the medical establishment has not really incorporated it into standard of cares, because they say we have not seen data that information about this actually produces more positive outcomes. But then at the end of the day, again, we have not really tried it in a systematic way, at least as far as I know. What do you think?
I think it's very much the same. Also, just getting that implemented in the general population would be a big undertaking. And then to have everybody and all GPs understand what it means, I think also would be quite hard. And if I'm being honest, probably because a lot of these things would likely involve also lifestyle decisions. If you had a higher risk for a specific thing, for many of these things, we don't have drugs. Like if you have an increased risk for Alzheimer's, what are you going to do different, except for maybe sleeping more, being more healthy, but that's a generally good thing to do. So I think it's very much, at the moment at least, it will be also very much optimizing when you're already at a high level of optimization. You could do a little bit more, at least focus your energy on a specific thing that would lead to likely the most results for you, except for those more extreme cases where there would actually be a much more immediate risk if you have very specific mutations. I think Peter Attia, a bit of a troubled person at the moment to talk about, but let's say in his book he mentions a few good examples of things. I think you are very much aware of those.
Yeah, I mean, one example that comes to mind is, so I have said it's not really incorporated into medical practice, but there is actually medical genomics that's already incorporated, at least in Austria or Europe in general, I think. But there it's the usual scenario of reactive medicine, that you have something has to go wrong in your case, or actually they already have expanded it. If you fulfill certain criteria that siblings, parents, or other relatives suffer from something that has a genetic origin, let's say cancer, and you fulfill certain thresholds, you can actually be tested. But this is then again genotyping, not whole genome sequencing. So they really have then a very clearly defined panel of genes that they check. So they sequence you, but they only check these specific locations for the variants, mutations that are associated with the trait in question. And these are panels of a few genes, like really 5 to 15. It's a very small fraction. In terms of protein coding genes, we have 20,000, just for context again. And in general, as we said, we have 3 billion letters that can be affecting to some degree the outcome. So this is really a tiny thing. I mean, it's a step in the right direction. And then also you have to kind of, in Austria at least, agree that you want to have this testing done. And also you will be, it will be very much in detail explained to you what the outcomes could be, and if you are in a psychological condition to actually take that, because you have to kind of prepare that you want and not have then an anxiety induced that might never realize itself. So you might never get the disease. Nevertheless, they have recognized that for this, as you said, high risk variants in genes, like for example, BRCA2 or BRCA1, they have a higher prevalence for breast cancer and ovarian cancer, that you then can actually check yourself if certain criteria are fulfilled. Yeah, exactly. But then on the other hand, yeah, go ahead.
I think in the end, an important part is how actionable are the things that are measured. And I still think the society where we're living in now, I think we are in a bubble of relatively health aware people, where maybe this makes even more sense to do, because other things, like we want to invest the time and energy to optimize and to make sure we can live our best lives in a way where we're healthy, we can be there for the people around us, and we have a lower chance of being in a way crippled by disease, or at least limited by disease. But for a lot of people, just the general advice of sleep more, eat more healthily, and move a bit more, those are the most important things. And then a polygenic risk score for Alzheimer's or heart failure are going to do very little for them if they are anyway already not following the first 3.
Yeah, you're right. I mean, that's then again this what we always discussed also on the episode where we discussed blood pressure, right? Who gets actually the notification, goes then to their GP, gets high blood pressure diagnosed, and then actually changes their lifestyle. This is definitely one thing. But then again, I always want to kind of push back a bit and say, okay, but at the same time, we are removing from however small the fraction in the population is that would actually act on the information. And we kind of stealing from them, although we have the knowledge and the technology and the capabilities, simply because they are not having maybe the money, the time, or simply the upbringing to be able and education to come to the same level of awareness as we are. In terms of, hey, I can go to an end consumer product, let myself sequence, look at the polygenic risk scores, and then devise an actionable strategy. That is a lot of steps that require a lot of interest, time, money, education that we should not expect everyone to have, but there might still be a proportion, however small, in the population that says, okay, if my GP tells me with quite a conviction that I have a high predisposition to, let's say, colorectal cancer, and the person just says, it is shown that if you move more and at least eat, 10 grams of fiber per day, which means eat for breakfast always, oats, you will decrease your chances to get that by, 150%. I'm just making up numbers. It's just these really actionable interventions where we know, okay, these lifestyle choices decrease the chance when I have a given high probability. And I would just hope that there are a few people who would then, without any background, just say, okay, my doctor told me that, and now I'm going to do it. I know it's kind of optimistic to hope, but at the same time, I'm always like, okay, if we don't implement it, we're kind of taking the chance away of people surprising us.
And maybe colorectal cancer would be an interesting example where also screening could be increased, or like these kind of things where medicine, or at least preventative medicine even, can do a little bit more. There, I think it makes sense. And screenings, or even in the future specific medications, maybe for you in your particular scenario, it would make sense to at an early age start with blood pressure medication or so. And I think there, the moment there is also screening increase potential for specific people where also the screening maybe has a risk to it, these kind of things would definitely help. And one thing we might think about is maybe family history is already enough. I think there's still some research to be done, but there was one good paper that I screened. I didn't read it in every detail, but it seems to be that for some reason, genetics and family history seem to have independent predictive capability, which I still don't fully understand, because you would think it should both be in there, but maybe our families are just not big enough to catch everything, or I'm not entirely sure, or maybe it's really these unique combinations that you get that actually can predict quite a bit.
But yeah, if family history is also predictive, like there must be an overlap in terms of genetics.
Yes.
But maybe the family history, now that you mention it, might have more explanatory power about lifestyle, because, family, everything you get taught as a kid comes, until you at least go to school or so, from your parents. And even if you're in school, I mean, I don't know if it was different for you, but I listened to my mom most of the time,
Yes.
like 90% of the time. So that means everything trickled down in terms of lifestyle, which is what I do regularly. And as we always preach, and what you do regularly compounds over time and also forms kind of your identity and how you do things. And maybe actually the family component encodes a little bit of lifestyle, although the genetic component there's zero lifestyle in it, right? Maybe that's why they have different predictive powers. I was very surprised when you said that now.
I also still don't fully understand it, but indeed that could definitely be part of it. And maybe we can also talk a little bit about our personal experience of sequencing and what we found out. So you did one type of genotyping or sequencing.
Sequencing. I did actually sequencing. Yeah, I did. Back then they were called Nebula Genomics. It's a US-based company, co-founded by George Church. Sorry, George. He is a big person in genomics, like a researcher who has basically, to some degree, invented some of the genomics technologies, and he's still an active researcher, and he's founded, I don't know, 3-digit number of companies already. It's insane. Anyway, this was for me like, okay, they seem reasonably priced. They have a co-founder I kind of have some trust in, and I went into genomics as a job, it's my occupation. So I felt like I need to get my whole genome sequenced for a reasonable price, and I actually did it for my 30th birthday, basically. Yeah, and the process was, you basically buy something. So you can either buy the sequencing and a subscription, or I bought back then the sequencing and that I have lifetime access to my results. And this cost around $500 back then. This whole package, also with analysis, and that they store my data and so on. And the procedure was quite simple. Then they send you from the US a vial and instructions, and then you basically scrub the inners of your cheek to get some epithelial cells, I guess. I mean, what do I get when I scratch my cheek? Yeah, so these skin cells basically, and you have to scrub quite hard. At least that's what the instructions told you. And then you put them in the vial, and then you send them off to the US. And basically, what they did, I think, they sent it then to China, and there the sequencing is done, and then the results are piped back. They process and analyze it, and then you get at some point, in my case, 6 months later, an email: "Hey, your results are ready, and now you can check out your results." And we can discuss the results. But first, what have you done?
So I did 23andMe, both. So I also did my parents just for ancestry. They didn't want to know anything about their health stuff.
Yeah.
So I just did ancestry just out of curiosity. It was very boring. I'm from the place where I'm from, and it could detect the province in the Netherlands where my genome was most likely located. So
what is a province in terms of size? Like, give us
a few million.
Okay.
Like 2 million, 3 million. I lived in the biggest province, but it was more of an area, but let's say 2 million people or 1 million people.
They could pinpoint it.
So my ancestors weren't very adventurous and stayed very much local. But for me, I did the health panel as well. And then so that's genotyping, not whole genome. I was actually part of a cohort in a study, and there I also requested my data. as a GDPR request. And that was also genotyping. So actually basically the same as 23andMe, a slightly different panel, but more or less. And then
panel means selection of locations in the genome, which is basically this catalog of which chapters do I want to copy or rent from the library, to keep with our metaphor.
Exactly. And then I did whole genome sequencing with Dante Labs, which I now did twice actually. So I did it originally back in 2021 or 2020. So this is whole genome. And then I tried again because I also wanted to do my parents now, to also store that data long term, just in case at some point they're not around, that I had that data, just out of interest, in case for whatever reason it might at some point be interesting to know. My last experience with Dante has been interesting. Let's say that a year and a half later, they still haven't processed my sample, and for a while they didn't respond to my emails. Now they say they are on it again, but I think I got a default email that said, yes, we will try to prioritize your samples. Will be hopefully done by June or May or something. But it was at the end of 2024 that I submitted my sample.
That's ridiculous. Especially when we said, now we can do it in 1 week.
Exactly. But I think they must have underpriced. It was a Black Friday sale, so I guess they got a lot of samples coming in. I don't know. That's beside the point. I just don't have my parents' data.
Okay. What's the pricing scheme of Dante, the logistics, and the package that you buy with that price? So for me, it was, as I said, 6 months it took from sample to results, and it took 500 euros. I get a lot of reports, and I have lifetime access. Although they also tried to revoke that, but that's another discussion.
So I paid, if I remember correctly, 200 euros per person, so 600 total for the 3, and you get basic analysis for health risks, and you just get that report, and then they try to upsell you even afterwards for new things panels that they, or no, new sort of risk scores that they can calculate. And
so reports. They give you a basic catalog, but that is fixed, so to speak, in terms
you get
of
a PDF
analysis.
more or less.
Okay.
And of course you can download your raw data.
Yeah.
So there's different formats there. In the end, I couldn't get the raw data of my original. So I think in the end, I got aligned filtered data, but not the raw sequencing.
Oh, no FASTQ files.
No. I think I think there were no more reads in there. I don't remember exactly, but I got my whole genome. I don't know if it was just a VCF or if details, doesn't matter. But the raw data that they were, of course, supposed to provide wasn't there anymore. But I think I got maybe aligned BAM files, they're called, doesn't matter. I've read different things, that some people got everything, some people didn't. It is what it is. I think for most people this will not matter, but I wanted to play around with the data. And even for me, like being familiar with this type of data, it was quite complex, just because there are different formats, and trying to align the 23andMe, the Dante Lab, and the third one I had, if I try to then make sort of an overview of my ancestry myself, they didn't perfectly align, which was a bit weird. But of course, they are all aligning to a different set of genes or mutations of genes, so there will be some variance there.
Especially if we know how complicated it is. That's why it's a job that requires studying a lot and then also having a lot of experience. And there are many parameters or reference files and file types involved in bioinformatics in general. So this is not a trivial thing. And also for them to provide it without the information of how they have processed it and how they derive then these downstream files, it's basically impossible for you to reverse engineer exactly what they did. And the likelihood that it maps to other downstream results is quite low, because if you just change one parameter, you get other results.
And I think
not fundamentally different, just when you say details that you cannot align them perfectly. Yeah.
Exactly. And I think one maybe important thing to realize here is that we didn't mention is one of the reasons it's cheaper to sequence now is because we have other genomes. We don't have to puzzle everything together anymore. We can sort of use that as a template to then put our data on. So it makes all the processing easier. Whereas for the first person that we sequence, we had to figure out everything. So just because we have that knowledge, we can use that prior knowledge to make the whole process easier. Basically have like a seed that we can use every time. And that is part of the reason why it's a bit cheaper now.
You could call it a reference.
Yes.
That's what we call it at work. It's a reference genome.
But what I got out of the sequencing is actually, so my ancestry was just pure interest. And then luckily I didn't have, for anything serious, an increased risk. So for me, it was kind of boring, but also good. Like there would have been the chance of getting these kind of more monogenic or really high risk diseases that might only manifest after 30. But that usually you would know your family history there. I think there's a better predictability, but I didn't have anything serious. Also no real increased risk for Alzheimer's or these kind of things. And all the data more or less agreed with this. But I also think still with these kind of reports that you get, they use a relatively limited set of more confident scores that we have. So there's the same things popped up, but I think they use the same basically reference database to predict these things. So it's not weird that they gave me the same prediction. They also should, hopefully. But of course, research progresses, things change. But so far, I'm good in general in terms of risk scores, even though, of course, from family history and from personal things I know, for instance, I have increased risk for high blood pressure. But this is also manifesting by the measurements I can take of myself.
So you see, but it also informs you in terms of you have the lifestyle that is ideal for cardiovascular risk, and then you see still a high blood pressure, then the likelihood that it's genetic because you kind of remove the variable of lifestyle and epigenetics, then that makes sense. It fits.
But there I think they're still lacking a good polygenic risk score that can more confidently predict, because for me it doesn't come out, but I also don't know if it's in there in the full panel. I think there was no proper high blood pressure risk polygenic risk score. What about you? What came out for you?
The cool thing is about Nebula, I'm not selling anything, and as I said, they have now, just to wrap up, the company doesn't exist anymore somehow. So they wanted to actually step back from this, hey, we give you forever all your data. And also what was part of the package is that whenever there's a new GWAS study, so a genome-wide association study that has found new locations in the genome associated with a trait, they calculate it for you and provide it to you. That was part of the lifelong subscription in a sense, right? What Dante wanted to upsell, I actually bought from, because I thought, hey, it's a good investment. It's once a measurement. And then for my lifetime, if there's always a new report, I get a new report. Well, it didn't really realize like that. So they said multiple times a month you get an update, which was not the case. But the reports increased to, I think, a number of more than 350 where they are now. But now they pivoted into another company. It's called DNA Complete now. And they only have subscription model, meaning no lifelong package anymore. And basically, they stopped providing me with new reports, but also warned me that they will delete all my data and all my reports. And I sent them, well, then give me all the reports that you have now from me. They never responded, but they're also not deleted. So I also have my raw data. I saved this on my own Google Drive. So just to wrap up the Nebula Genomics business model and how this is not optimal, but nevertheless, I think I had a better experience than you with Dante, because I have all my raw data, I have 350 reports, and it took 6 months and not. We don't know how long it will take, actually. So they cover also a lot of things. I will only talk about 3 categories, and within each only 1 or 2 examples to not make it too long or boring. But from the ancestry perspective, they do deep ancestry, which I don't want to go into. It's really like, as you said, which province and how many percentage points, or do you have a relative maybe in Poland or so on. But what they also give you is like Genes K36 calculator, they call, and they give you then percentages of your genome comes from this part of the world. And my top is 16.4% is Iberian, so Spain and Portugal. And I'm the whitest of the white, which is really funny. And then I already get like 15.7% of East Central European, which I think is much more, where most of it comes from, and then some Western European, Italian, and then we're already in the realm of 70%. So I'm quite Central, East, West European. In terms of traits, I also have increased high blood pressure. But I'm also more likely to be taller than average, which is a lie. I'm smaller than average, and I'm shrinking, apparently. Recent tests have shown that I'm smaller than we discussed before. But interesting, I have likely to have an average muscle strength, but you're more likely to have increased muscle volume. So, yay! So at least from the genetics. And also that I'm nearsighted is also higher likelihood. But again, it's probabilities, as we have discussed.
I think some things can be predicted very well, like probability of a light or dark eye color, attached or detached earlobe. Some of these things are relatively good predictions.
Skin color as well, to some degree.
Skin color, like from dark to very light, for sure. Like if you're basically you can with almost 100% certainty detect if somebody's fully from a European ancestry, like Middle European or Central Africa. This you can just with 100% certainty say.
Exactly. And then there is these reports, and there I have a whole library of 350 reports. And what they report is they give you a percentile where you are at with a certain association of a certain trait. And percentile is that so if you take all the data that they have and put it into 100 buckets, and I'm, for example, in the 100th percentile, that it means I'm in this bucket where 99 other buckets have less of a risk, so to speak. So I kind of went through them and sorted them now by this score, by this percentile score. And also I have a selection of things that I find interesting because they're actionable. Back to your point, why we don't have it so strongly in medical practice, ingrained as well, because from a clinical perspective, it's always cool for the information, thanks, but what do I do now? And if a doctor doesn't have a clear strategy by guidelines and research, they actually don't really care because they cannot do anything about it, right? It's not actionable for therapy or an intervention, or as you before said, it's a very good point, screening could be increased.
Just as a side note, technically there is no 100th percentile. So you're not if you're in the 100th percentile, it might be a roundup from 99.5.
Yes.
But just for the people very aware of statistics.
Not that we kill a statistician somewhere.
Previously you also said 150% decrease of risk, and I think don't know if a decrease of risk can be 150%. But let us know in the comments below if this is possible.
If you want to discuss hazard ratios and how to decrease by 150% or whatever. Yes. So for example, one is that is I think known by a lot of people. Lipoprotein B levels, and there I am in the 100th percentile, so between 99 and 100. And that means I have a genetic predisposition for higher ApoB lipid proteins, and these are the proteins that are containing LDL. This is this low density lipoprotein. And every LDL molecule circulating has one ApoB protein on top to mark it, so to speak. So this is really a clear predictor of cardiovascular disease, and there I have an extremely high predisposition. And this, for example, could very much inform me, which it does, that I should look out for my actual ApoB levels, right? Because this is just a genetic risk. So I always ask, if I do my annual physical or so, please not only check the LDL, I want to see the ApoB values, and then I want to have them in a good range. That is not like by whatever reference we're using.
Do you know what percentile you are actually at in terms of concentration in your blood compared to the population?
No, I don't know compared to the population. I have no idea about ApoB in the population. I just know I'm still in reference, I'm in the green, but somewhat in the higher end of the green, definitely. But I'm very happy about being in the green, and I don't know if this is of interest, but it seems that if I eat saturated fat, I have a very strong response to this translating to high LDL or high ApoB carrying molecules, which is not true for everyone. So there is again genetic predisposition. But if you're aware of that, then you can translate it to, okay, where is a lot of saturated fat? Saturated fat, just as a mental reminder, is always the fat that is solid when it is in room temperature, so coconut fat or butter. And in foods, it's in animal products in general. So coconut is not an animal product, no, but all the others, the fat usually from animals is saturated fat. So if I decrease my consumption of butter, coconut, and meat, I actually see that this helps with keeping the ApoB low.
So this was an example of where it was actionable for you.
Exactly, yes. Something that's absolutely not actionable, but interesting to know is I'm in the 100th percentile, which doesn't exist, of testosterone level. Okay, good to know. Could be useful, I guess. Maybe, one more is, for example, thyroid cancer, which is also to some degree actionable, because if you go to your annual physical and they do sonograms of your thyroid, then you can just ask him, hey, please look really carefully because I have a genetic predisposition. But then maybe one fun, the final one is, I shared this before when I got my results the first time I shared them in the lab. I have a 93rd percentile for the trait called systemizing. And the description is, systemizing is a trait with describes the tendency of an individual to search for or create structures in the physical environment or mentally. I guess no one was surprised, but it's encoded apparently. So
Interesting.
These are the results that you could glean from that are ranging from curious, interesting, or actionable, but definitely make sure you are in a state of mind in general, or you have arrived at a level that you say, okay, I have the capacity to understand that these are probabilities and that they give me sometimes the agency to do something about it or motivate me actually to keep doing something. For example, my cardiovascular risk motivates me to lower my saturated fat consumption and increase my cardiovascular or also strength training. But at the same time, there are some risks where we don't know what we currently can do. But in the future, if you see the risk now and 5 years from now, there's a really good intervention. And that we know, for example, that as it has been unfolding now for neurodegenerative diseases, have a good omega-3 index and so on, then you're like, oh, okay, then I can include it into my protocols or whatever.
Yes, and maybe talking about the future, that will be a nice segue to the next section to talk also about future outlook and how to implement it for society, potentially, what your thoughts are on that. But first, talking about the future or near future, if you enjoy our content, if you could press that subscribe button, that would be amazing. Either if you're on YouTube, it's subscribing, or also subscribing, or I don't know if there's different following on different platforms, especially things like Apple Podcasts, Spotify, and also to leave a review if you enjoy it, because that helps us reach more people, which will hopefully be also helpful for those people. So if it was useful for you, it would be great if you could do that, and also let us know in whatever comment section you're in what we could change and improve. So you might have noticed recently that we're more focusing on a single topic per episode, to make it a bit more clear what the sort of episode is going to be about and have more structure in that. And that was based on comments also from all of you. So that would be amazing. And thanks also for watching. And hopefully we can make this better in the future. And talking then about future again, how would, in your ideal scenario, say we could both sequence everybody that is alive in Austria right now, and also we could potentially, there's maybe some data privacy issues there, but sequence everybody that gets born the moment they get born, basically. Yeah. What do you think about this, and what would be a realistic and ideal setup for you?
Yeah, I thought about this a bit, and there are initiatives that I find really cool, mostly in the US, that try to do this exactly like I would outline it, but I've come up with this in parallel to some degree. So there is like Bayesian thinking, Bayesian statistics, where you have a prior probability and a posterior probability, and you can always update your probabilities. And I think that's how it would be ideal to think about all of this. And the initial and prior probabilities we already have some, because we have epidemiological data of incidence of certain diseases given some demographics. So for example, women are more affected from autoimmune disease, and men are more affected from cardiovascular disease. Also, cancer is more in men, and so on and so forth. So we know simply by one variable already that there is a higher likelihood. And we have a lot of variables from everyone already in the current clinical paradigm. So there would already be an easy way to get a probability just based on your demographics. And this, like the demographics really that exist basically when you're born. So really the basic demographics. And then if we have a whole genome sequence and then have really meaningful predictive prognostic polygenic risk scores, we could already combine this. These are both kind of fixed points. So if you're a woman and if you're born in Austria, this is kind of fixed, right? And if we combine this now, always with the lifestyle, this is very difficult, but there we can basically incorporate wearables and trackers, which have a continuous nature of giving us, for example, information of how much a person moves, in combination with your annual or regular physicals, your tests that you do with your GP, with your general practitioner, as terms of the standard blood panel tests. This is already capturing the snapshot and actually what your lifestyle and your environment and your epigenetics lead to. So you have your genetics, your demographics, and then you have your epigenetics and lifestyle that continuously change, and you update your probabilities in this Bayesian model. This would directly inform them in this ideal scenario, your general practitioner or your physician, to tell you, hey, we're going towards a direction where there's really an increased chance for, I will use it again, colorectal cancer, because also for the listener, colorectal cancer has a high lifestyle proportion. So this is high, there is a genetic predisposition, and then it can be very much addressed with lifestyle. And what colorectal cancer also has is a screening approach. And this then goes full circle to what you said. The clinician actually has an action that they can perform. They can say, hey, these lifestyle changes would be helpful. These are the markers what we see. But also because you're currently moved with your probability into, 80%, we now start screening you with, 35, 40 instead of 55. Because the screening is also informed already by demographics. The screening numbers are not made up randomly. They check their demographics and then they look, okay, when should we start screening, in which cadence, in which interval? And maybe you should start earlier and more often because the risk benefit ratio speaks for it. So this would be not talking about newborns, just this. If we would have the sequence of everyone in Austria and could implement this, would already, I think, increase the health and especially the preventative aspect of our healthcare system a lot, and would potentially avoid or push out this chronic debilitating, right?
Yes.
Debilitating phase of your life, or compress the morbidity phase to only, 5 years instead of 20 to 30. So this would be my approach or vision. Obviously, there are many hurdles, one of which you already addressed. Security would be a bit of a problem.
Yes.
Who owns the data and so on?
I think both owning the data, but also access to the data and what the consequences of this data are. So I think in Europe, we tend to have a bit better protection for our data than America. And also, I'm talking here about the Netherlands, but there's rules that, for instance, say insurers cannot refuse you based on your past or things they know about you and your risks. Even if you already have a disease or if you're at high risk for a disease, they cannot refuse you. And I think there we need to be very hopeful and careful that governments take charge of this, and it's not private companies that take charge of this, because the moment private companies have that data, they could give access to insurers or other providers of healthcare or something that would in the end influence the quality or price of your healthcare. And I think there's a lot of danger. Plus, and this is something we cannot really solve, is, you mentioned this in the beginning, some people don't want to know. They would live a happier life, even if it's potentially a bit shorter, or maybe they have a longer period of disease. They would still live a happier life not knowing. And this is hard. If you start this kind of protocol at, let's say, 23, when maybe your brain is more fully developed, or 18, when you're technically, no longer a minor.
Yeah.
Then you make at least an informed decision, but that decision might also change over time. If we then move it to newborns,
Yeah.
it becomes even
More
harder.
difficult, yeah.
Of course, we already make a lot of decisions for a newborn also that have long-term consequences. So it's not completely different, but then there we should be really careful with data privacy. And it could even be that say we based their treatment or how we raise them in a way, very specifically with regards to healthcare. And the parents can know, but the newborn just doesn't know, or whatever, even if they're 12 by then, they don't really know why maybe they're getting a specific medication, or maybe why their parents focus more on certain foods. And then they get the choice at a certain age to know more. But I mean, this is very much practical implementation. That's very important.
But we already do this to some degree, right? But nowadays it's very reactive. So our medical system is built on a reactive nature, that if someone has high fever, a cough, and is 70 years old, so there might be a case of lung infection, pneumonia, which is can be lethal, especially the older the patient is. No doctor in the world would not say, oh, do you actually want to know what you have, or should I not tell you because you feel better? No, every doctor would say, here's the case, you have pneumonia, that would be the treatment. Obviously you can reject the treatment, right? But also no one in their right mind, in the current mindset, would then say, no, I don't take the antibiotics, I kind of want to meditate it away. I mean, I'm talking about the average person. We know all that these cases exist. But now obviously it's very different. It's now something that might happen in the future. Obviously, also a diagnosis from a doctor, given a presentation of a disease and a treatment course, is also just probability. But the probabilities are just more higher, right? If there is pneumonia, there is high fever, there is a reason to act, and there is also immediate feedback. In the case that we have been sketching there, the feedback happens 30, 40, 50, 60 years later, maybe, or even has not a discrete event, but just a lifetime quality of life is 10% better, whatever that means, right? You are more agile, you have more energy, you simply have more agency over your life, or you are happier, whatever that means. I don't know. That's then difficult. And I think that's the big difference, that we have longer timelines and more nuanced changes, and not this kind of, okay, I'm missing a limb. Yeah, let's close the wound, basically. But over time, medicine has developed in this case. Like just this example, there's an accident. Yeah, we patch up the wound. But then now we have diseases which are already not so acute anymore. And now we have chronic diseases, which are really not acute, right? And they develop over a long time. And I think we have to have, as a society, a mindset shift towards thinking more about long term. I mean, now we can start with climate, but we don't, because we wanted to focus in our episodes.
Yeah, and I think there it's really interesting that, to my surprise, sometimes governments are able to implement this, because my thinking would be, there's a 4-year election cycle. Exactly. Why would they focus on energy? But I guess there is still that direct effect of people getting sick from heart disease or something that could have been prevented. So that still puts some pressure to prevent this in the future. That's one of the reasons why probably cancer gets so much money, because it has such a direct effect on so many people, and also often on people that are generally healthy and that all of a sudden then get a disease. So that sort of urgency and unfairness helps us maybe shape for certain diseases prevention much better than for others. Like smoking rules were not always popular, but still they were implemented in Europe quite well in the end, I think.
Yes, definitely. And for the newborns also, I mean, if your pediatrician tells you as a parent, okay, we have now seen that your kid has asthma, like at a very early age, this sometimes happens, right, that they have just asthma, then there will be, for sure, I mean, you would be considered a bad parent if you don't address this, right? And this also, again, it's something immediate, because your kid somehow has trouble breathing, coughs a lot, you bring them to the pediatrician, and then they tell, okay, yeah, there's a case of asthma, and you should look out, especially during these seasons, and you have to take this treatment. And everyone would say, yeah, that's what a good parent does. But then where does it stop? If we have this kind of tool and we have this kind of predictive power, and you really see already in the data, okay, it's so much easier to educate and bring up a child with certain lifestyle choices and habits that are then helping it. And then at 18 or whatever we take as a cutoff, like full prefrontal cortex development at 25 or legal being mature enough to vote and drive cars and so on, to give them the chance, okay, now you can have access to this data that, at least for us, as parents, we use to make the decisions. So that's I think an ongoing discussion. And people have been dreaming about that for a long time. Maybe as a closing remark, already in 2013, there was a paper that was titled The $1,000 Genome, an Ethical Exploration. And then the chapters in this paper were really what we have been talking about. They start out with, hey, obviously we can do research on the genome, but diagnostic testing, screening of newborns, and prenatal diagnostic testing and screening. So really before birth is prenatal, right? Screening of embryos. And then what does it mean if we have this $1,000 threshold crossed? And this was 13 years. Sorry, my calculation was wrong. So we're in the year 2026. 13 years ago, they already had these thoughts, and now it's not cost prohibitive anymore. Now it's really, I think, regulatory prohibitive in a sense, and we have to have the will to address change and also do some more research. As we said, genomics is quite a young research field from the natural sciences perspective.
Yeah, and I think say we implement more and more of this, as that happens, we also learn what works, what doesn't work. And that, I think those practical things are also in a way research to get it implemented in a way that has the most positive societal effect.
Totally. Yeah, so today in summary, I think we started out with this announcement, $100, and then we also put in perspective what is actually the difference between whole genome sequencing that is now so cheap and what 23andMe basically sold, called genotyping, and these different products. We introduced polygenic risk scores, how they're used, how they connect back to what we discussed before, genome-wide association studies, and also what our experiences were with 3 different providers. And you saw also the range of cost and the results that we got. And yes, we discussed a bit the future of personalized genomics, and we would definitely be interested in what you think about this topic in general. Would you want to know your polygenic risk scores? Would you act on them? Would you say, okay, no, this is too abstract? Did we explain it well or not? Yes, please leave reviews, please leave comments. We need this for the platforms, and also that we get some signal back if we do a good job.
Yeah, and in general, for this particular episode, I would be interested in if you've had some kind of sequencing or genotyping done and how you use that data, or if it was purely just, I want to have some interesting, fun data. Well, thanks so much for listening, watching, and see you in the next episode.
Be your best N of 1. See you next time. Bye.